Your host for the morning session, Professor Gregory Fulcher.
Good morning, ladies and gentlemen. It's a great pleasure to be able to welcome you to this important symposium at the IDF, and in particular to welcome you to Melbourne, Australia. This is one of the cities that we love most in this country, and I'm sure you're going to enjoy it enormously. You won't have a chance today to see any of these iconic sites in Melbourne, but once the symposium's over, once you've seen all the Novo Nordisk abstracts and been to the important scientific sessions, I hope that you get a chance to hop on the free tram that takes you all the way around the city, and you can hop off and see some of these sites. And I won't spoil it by telling you what they are, but no doubt you will see most of these things before the end of this meeting in Australia. We do have a technical issue with the slide advancer, I'm sorry. Okay, so here we are. This is our little island. We've got something like 23 million people that live in Australia. We've got about 3.6 million of those have got something wrong with glycemic control, whether it be diabetes or pre-diabetes. Most of this country is actually desert, so don't be tempted to walk off into the middle of Australia. You won't find very much there. Of these people that we have in Australia, we also have about 3% of our population is made up by the Indigenous people, the Aboriginal people of Australia. And I'm going to come back and show you a little bit of information about those people a bit later on. We have over 10,000 deaths annually that can be attributed to diabetes. And this fraction of our population, there's a little bit of variation on this, but somewhere between 7.5% and 10% of our population have got diabetes. As you'll see when you're walking around Melbourne, we have a wonderful ethnic mix in Australia, but as more people are coming from Asian countries or from countries with high background prevalences of diabetes, then the prevalence of diabetes in our country is also increasing. Most of our costs associated with diabetes relate to inpatient care. You can see a breakdown. I'm not going to go through these in specific detail, but a lot of the inpatient care of diabetes can be avoided by good management of blood glucose, obviously, and all of the other factors that contribute to the causes of people coming into hospital with diabetes. And these costs, as you know, and this is not different here than it is anywhere else, are increasing, and you can see from 2005 to 2008 that when you put all of these costs together, and these are the costs in millions of Australian dollars, that this is an enormous cost for a community, but of course importantly for the people in the community, it's an important cost for them in terms of the way in which diabetes affects their lives. This is our Indigenous population, or some data relating to our Indigenous population. And like the Indigenous populations in many countries, they are worse off with the arrival of the Western civilization to their country. And you can see here that if you compare the rates of diabetes in Indigenous people compared to non-Indigenous people, firstly, there's a similar age increase. And of course, this is a population that's got a decreased life expectancy compared to others in Australia, but we have around 3 times the prevalence of diabetes in this group of people compared to the rest of Australia. And this, of course, is something that we're all very passionate about doing something about. We have a long history of some policy development around diabetes, and I think that many of you will be going through or will have gone through this same sort of exercise in the countries that you come from. In 1989, the World Health Organization passed a resolution to set up a National Diabetes Action Plan, and in 1993, the Australian Diabetes Society did a fair bit of work with the incumbent government to ensure that diabetes was on the national agenda. And in 1996, diabetes was made one of our top 5 health priorities. And of course, that's very important because that focuses attention on this important condition. The National Diabetes Strategy and Implementation Plan included a range of goals for the management of diabetes. Improving the capacity of the health system to prevent and treat diabetes, and Australia is a little different to other countries in that we have a state health system which is largely to do with hospitalisation, and we have a federal health system which is largely involved in the community aspects of health management. And so this very clearly sits in primary care strategies to try to encourage activities that will delay the progression to diabetes and also aid in the early detection of patients with this condition. We want to improve the health-related quality of life with people with diabetes, reduce complications and also reduce premature mortality, and of course focusing on the management of diabetes in pregnancy, and there's all sorts of issues around that. And you would be aware that we've got new guidelines, and those guidelines will increase the number of patients that we have to deal with in our hospital system. And of course, we want everyone to know about diabetes. We want them to understand the implications of their current lifestyles and activities in terms of the likelihood of that leading to a progression to diabetes. So for Australia, it's one of our foremost healthcare challenges. The costs of diabetes is rising, and there are new strategies that are currently being implemented to raise awareness and improve the treatment of people with diabetes. We have a number of speakers in today's symposium. They come from countries all around the world. We're very grateful that they've made the effort of coming this very long distance to Australia. These are the speakers, and these are their various disclosures. There is going to be the opportunity for all of you to participate. We want this to be a very active symposium. We want the audience to be involved in it. And one way that you can be involved is to ask very challenging questions of the panel. And we're going to do this today via question cards. So there will not be roving microphones, but there will be question cards. The gentleman on my left is holding those up at the moment. They are in the pack that you would have got as part of your introduction to the symposium, and you can fill those out and you can pass them to the assistants who will bring them to the chairman of the question and answer session. It's very important that you evaluate this program. Novo Nordisk are very interested to know the things that you find interesting, the things that you would like to see in future symposium, and any criticisms that you have of the way in which the symposium is run, or the program itself. They would love to hear that will assist in the ongoing improvement of these programs as they're conducted in various places around the world. I'm sorry for these technical issues. So, I think what we might do is I might actually introduce our chairman for this morning's session, Jiten Vora. Jiten is going to lead you through the symposium and will be introducing each of the speakers and linking together the story that we're going to be telling about the role of some of the new therapies in the management of diabetes. Thank you very much for your attention, and we will now get on with the main program. Thank you. I don't know what these things do. They're just useless. Okay, thank you.
Well, good morning, ladies and gentlemen. As you can see, my name is Jiten Vora, and I come from the city of Liverpool in the United Kingdom. And I would like to add my welcome to you this morning, to the morning session of the symposium, which it is my pleasure to chair. Now, what we're going to do for you this morning, ladies and gentlemen, is we're going to tell you a little story. The story is going to start off with the development of insulin, and we're going to move through and present to you some new data around pharmacology and pharmacokinetics of new agents. We're going to give you some ideas about clinical use of these new agents, and also potentially a newer set of combination. So my role in the next few minutes is simply to present you with an introduction to this story. So here is a story starting off with the discovery and development of insulin, moving on to prolongation of its action with Hagedon, the development of new insulin analogs in the form of detamir and glargine, and most recently, the development of a state-of-art preparation, insulin degludec. We will also present to you some data around combinations of the newer agents that have recently become available. So the role of a basal insulin is well established in clinical practice, but there are some characteristics and some properties that you and I as clinicians desire in a basal insulin. Firstly, it's the ability to control fasting glucose with a single daily injection, and thus having a long duration of action. Secondly, we want to achieve glycemic control with a lowered risk of hypoglycemia, and this means a flat, peakless time-action profile. From the patient's perspective, we want less variability, more predictability of their glucose profiles, and therefore we should have a basal insulin which has less day-to-day variability. And finally, what is crucially important to the patient is a degree of convenience and flexibility. And this would, of course, relate to a long duration of action. So with this background, we have to ask ourselves, what do our current basal insulin preparations provide? So here is NPH insulin, and this curve represents glucose infusion rates to maintain euglycemia in a single individual who was administered NPH on 4 consecutive days. So it's essentially evaluating the pharmacodynamics of the NPH preparation. There are a number of points to note here. First of all, the 8 to 10-fold variability in the glucose infusion rates. Secondly, the considerable peak in shoulder that you see with the NPH preparations. And thirdly, the variability in its duration of action profile from anywhere between 12 to 16 and 18 hours. The development, of course, of the newer insulin analogs in the form of insulin glargine resulted in a considerable improvement. So there was a degree of reduction of the variability and an increase in the duration of action. The same was true, of course, with insulin detamir. Again, some reduction in the variability. Now, what we want as a clinician for our patients in terms of a basal insulin is a flat, smooth profile without a peak, with an ultra-long duration of action and a reduced variability in its pharmacokinetics as well as its pharmacodynamics. So, of course, the variability is particularly important when we consider fasting plasma glucose. Here's a schematic representation of a patient's fasting plasma glucose over a period of 30 days or so. You will see that there's a considerable variability from the peaks and the troughs, despite the fact that the average fasting plasma glucose is around 7 millimoles. An attempt to achieve euglycemia or normal glycemia by reducing fasting plasma glucose to a level of 5 with this profile of fasting plasma glucose levels would mean that the patients would have a considerable period of time in the hypoglycemic zone. So their excursions into hypoglycemia would be increased. If, on the other hand, we have a preparation that provides a fasting plasma glucose with less variability, as depicted here, you may see that the average is still around 7 millimoles for fasting glucose. However, attempting to improve glycemia in such a patient would mean that you would achieve this improvement in glucose control, fasting glucose control, without an increased risk of hypoglycemia. And there is, of course, data that supports this. Here is data on glucose variability and incidence of hypoglycemia in the DCCT study in patients with type 1 diabetes. So here we have tertiles of glucose variability. Whether you were allocated to conventional treatment on the left or the intensive arm in the study on the right, and you will see that as tertiles of variability increase, as the variability increases, the incidence of hypoglycemia increases in these patients, whether they are on conventional treatment, i.e., their HbA1cs were not very tightly controlled, or if they were intensively controlled with appropriate HbA1c levels. The same data is also available, or similar data is also available for patients with type 2 diabetes. This is quite a busy slide, so let me take you through the axes. Here, we have increasing glucose variability. Here, we have declining HbA1c levels. And on this axis, we have increasing frequency of hypoglycemia in numbers of events over a 48-hour period per person. You will see that for any band of HbA1c, here above 8.5, the incidence of hypoglycemia increases considerably as the variability of glucose control increases. The same is true if your HbA1c is declining to between 7.5 to 8.5. But the most important data here is that if you attempt to achieve what are our normal targets for HbA1c, around 7 to 7.5%, there is an exponential increase in hypoglycemia with increasing variability. So variability is important in both type 1s and type 2s, and we've schematically discussed this in terms of fasting plasma glucose. Now you know that one of the questions that we are all frequently asked is, do the newer insulin analogs protect or have a benefit in terms of reducing hypoglycemia? The issue has been made difficult by the fact that in regulatory studies, patients who have had episodes of hypoglycemia are frequently excluded. So that phase 3 studies invariably have patients who have a low absolute rate of hypoglycemia. So here is a study that was recently published or presented at the EASD in September, looking at a population of patients with type 1 diabetes, and to be enrolled into this study, you have had to have 2 episodes of severe hypoglycemia over the preceding 12 months. In the prospective arm, you were then randomly allocated to either receiving human insulin and NPH insulin, or fast-acting and basal insulin analogs. This is a randomized, open, but with a blinded endpoint trial. And what it showed us was that for severe hypoglycemia, patients who received the analogs had a 30% rate reduction. So in absolute terms, this means that there is a reduction of half an episode of severe hypoglycemia per patient per year when transferred to insulin analogs. Of course, majority of these episodes that patients experience are mild. So did the study demonstrate any data with mild hypoglycemia? So firstly, you will note here that there was 1.05 episodes per patient per week when they were allocated to the NovoRapid and insulin detamir arm. And those who were allocated to the human insulin and NPH had approximately 1.12 episodes per patient per week. So as an overall reduction of hypoglycemia, this was reduced by 6%. But there was an impressive 30% reduction in episodes of nocturnal hypoglycemia. So this is a study in patients who are exposed to, likely to have episodes of hypoglycemia, particularly severe episodes of hypoglycemia, in whom transferring them from human insulin and NPH to insulin analogs resulted in significant clinical benefit. So then let's move on to what we might hear through the course of this morning. You will hear considerable amounts of data on insulin degludec, and the data on the insulin degludec has already been published in terms of clinical pharmacology, demonstrating that it has a duration of action of somewhere of the order of 42 hours. And then we've also looked at the pharmacodynamic effects of insulin degludec, demonstrated by the glucose infusion rates that patients require to maintain euglycemia. Here you have data that shows you that the glucose infusion rates remain flat over the course of 24 hours. You will also hear a significant amount of data around the phase 3 studies. Firstly, these will look at once-a-day injection of degludec in patients with type 2 diabetes compared with insulin glargine, or using degludec in a basal bolus comparing with glargine or insulin detamere. Or you will also hear data showing you the benefits of degludec in flexible administration. And finally, a comparison with the oral agent sitagliptin early in the disease spectrum of patients with type 2 diabetes. Majority of these studies have now been published in high impact factor peer-reviewed journals. And this represents the fact that the phase 3 trial with degludec was indeed a global trial, including 11,000 patients across the world in the program. There is some data that will be relatively new through the course of this morning, looking at extensions of the once-a-day as well as the basal bolus therapy trials. So hopefully through the course of this morning, we're going to show you data around insulin degludec in controlling fasting plasma glucose with a once-daily injection due to its long duration of action. We're going to demonstrate to you that there is a reduced risk of hypoglycemia despite achieving same glucose control, and that's due to its flat action profile. We will also try and show you a predictable glucose profile, glucose-lowering action, due to a reduction in variability with insulin degludec. And finally, you will see considerable amount of data in flexibility of administering insulin degludec. So this is the story that we were going to tell you through the course of this morning. Now, the next chapter in this story is to examine the bioengineering behind the development of these new products. And to give that presentation, it is my great pleasure to introduce Peter Kurtzels, who is the head of research and development at Novo Nordisk. Now, ladies and gentlemen, you need to pay attention to this presentation. If there's anybody in the world who knows anything about protein engineering in relation to insulin, Peter is the man. So, Peter. That's probably better. Yeah. So, Peter, we as clinicians always have demands on scientists and bioengineering researchers and laboratory workers. We want longer insulins, we want faster insulins, we want less variable insulins. Can you deliver?
Absolutely. We will try to deliver. You know, I've been part of trying to deliver for almost 25 years now. And it's been a journey, a very exciting journey, and I'll share some of that with this audience over the next 20 minutes.
Excellent, Peter. The floor is yours.
So thank you very much. And as I said, it's been an interesting journey to work with designing new insulins, new GLP-1s, new protein drugs for so many years. And what I'll try to do in this presentation is to kind of take you behind the scene and understand some of the basic science, particularly around insulin degludec. But before we get into insulin degludec, you know, the insulin molecule is a protein. Like GLP-1 is also a peptide or a protein. And most proteins in nature are not really in the native form optimized for pharmacological use. They may be too short-acting. They may have other deficiencies, such as being non-stable in aqueous solution, and therefore often we would need to optimize the molecule by an analog approach prior to pharmacological use. And in doing so, there are various aspects to consider. So when we do new analogs, we have to preserve efficacy and safety. We have to engineer into the molecule the right kinetic properties, so to have the right time-action profile. And we also need to make sure that pharmaceutical properties, such as stability, such as the possibility to express in high yields, to give a good pharmacoeconomic situation and so on, all of those parameters have to be taken into consideration when we choose the final analog for clinical use. And in doing so, we can take various approaches. If we are working with a protein with which we have very little basic understanding, we could take a random mutagenesis approach, as illustrated in this slide. So we would basically create analogs by a random approach, and then select via various assays, that could be stability assays, it could be efficacy assays, what protein has been improved, and then we could end up with a molecule that would be relatively good in as a clinical candidate. But for molecules such as insulin, we know much more when we start, and therefore we can take a much more rational design approach to designing the right properties into the insulin molecule. In this approach, we take, for example, a computer-aided design. We utilize all the knowledge that we've created for years of that specific protein, insulin in this case, and following then an optimization protocol, we'd also end up with an improved protein. And insulin degludec is an example of such an improvement. So what do we know about the insulin molecule that can guide us in designing a new analog such as insulin degludec? Well, first of all, we have a very detailed mapping of what dictates receptor recognition. And it's illustrated in this slide that we know that there are at least two major receptor binding sites of the insulin molecule, site 1 and site 2, illustrated with the two colors here. And we also know what residues of the amino acid sequence, as illustrated here in the bottom, are important for really governing receptor interaction. We also know that another parameter is very important for insulin stability and also for insulin time-action profile, and that's the property of insulin self-association. And we have also, over the years, been able to map what residues are important for insulin dimerization or for formation of the insulin hexamer, which is a natural storage form of insulin in the pancreatic beta cells, and also typically the most stable form of insulin in the vial or in the cartridge, so that we can have a stable aqueous solution. And we also know what part of the insulin molecule is really governing insulin self-association, again here illustrated with the color codes in the bottom. So when designing a new analog, we really want to keep the receptor binding surface as intact as possible. And when working with basal insulin analogs, we'd also want to keep the insulin self-association surfaces as preserved as possible to make sure that we have a stable pharmaceutical preparation. And that's why when working with basal insulin analogs, the main effort has been in the C-terminal part of the insulin B chain that's out here in this region of the molecule. And you can see that both for insulin glargine, also for insulin detamere, and for insulin degludec, the major modifications to the molecule has been made in that particular region of the insulin molecule to preserve efficacy and also to preserve stability of the molecule. But let's look a little bit more closely at insulin degludec. When we set out to work with insulin degludec, we really had the ambition of creating a new mechanism of prolongation, because we were saying to ourselves, well, we need to come up with a new invention to have a true flat action profile and to have an even longer action profile than we know for the first generation basal insulin analogs. And to that end, the most common property to work with to prolong insulin action is to work with the property of molecular size. And to increase molecular size and thereby prolong insulin action, you can take various approaches. As illustrated in this cartoon here, we could take the approach of a chemical conjugation. That could be with a PEG, with an X10, with carbohydrates. Could also be by a fusion protein with either an Fc portion of the antibody or an albumin molecule. And a fusion protein would be an alternative way to pegylation, for example, to increase molecular size. But what's shared between these various approaches here is that you actually do quite a dramatic change to the parent protein by coupling with those kind of very large molecular substances. So we've taken another approach in Novo Nordisk and make much more subtle modification to the insulin molecule by a mechanism we call protein acylation. And by acylation with fatty acids, we can preserve almost completely the insulin structure. We only make very small modification to the insulin molecule, but we can engineer, in the case of insulin degludec, the possibility to self-associate into a multi-hexameric state at the site of injection, and a dihexameric state, as I'll come back to and explain in more detail, of insulin degludec in the cartridge or in the pharmaceutical formulation. And then, following over time, following release of zinc, we'll have a release of the active degludec monomer for absorption and action. And let's go into a little more detail about that, but let's first look at the insulin structure, the degludec structure. In the insulin degludec molecule, we made no substitutions in the amino acid backbone. That means what is ultimately recognized by the receptor is the same amino acid sequence as you see in human insulin. We did delete residue B30, which is not important for insulin receptor recognition. And then we attached at position B29 a side chain composed of a gamma-glutamic acid spacer and a C16 carbon diacid. And this diacid here has a specific property of facilitating association of the degludec molecule into multi-hexamers in the absence of phenyl, as I'll explain a little bit later. So this is really the molecule that is very close to human insulin, actually identical to human insulin in its amino acid sequence, and then just carries that modification at position B29. But let's try to look into a little bit more details of the science around formation of the multi-hexamers at the site of injection and the dihexamers in the vial. And to understand what is going on, we have to understand that the insulin hexamer can be present in various forms. First, this form here, as illustrated on the right, is the so-called T6 configuration of the insulin degludec molecule, in which the hexamer is open in both ends, and you have the fatty acid tail here radiating out from the ends of the hexamer. And in the central core, you see with yellow here, the zinc atoms that keeps the hexamer together. So in the absence of phenyl, the insulin degludec molecule or hexamer here is open in both ends, and you have the fatty acids here radiating out. We can show that in a spectroscopic analysis, where you see that here in the absence of phenyl, at no phenyl, we have the T6 conformation present of insulin degludec. But once you start to titrate in phenyl, you'll see that there's a conformational change going on into the stage what we call the T3R3 conformation. And let's look at the T3R3 conformation. In that conformation, in the presence of phenyl here illustrated with blue, we have the insulin degludec hexamer open in one end, but closed in the other end. So this is a special configuration that we have for insulin degludec, open in one end and present and closed in the other end, but still with zinc present in the central cavity here to keep the hexamer together. And what's so special about this T3R3 conformation is that it allows formation of dihexamers in the vial. So in the presence of phenyl, we have two insulin degludec hexamers here coming together via the open ends, and then the fatty acid side chain can reach into the neighboring hexamer, into the zinc atom in the neighboring hexamer, and then keep these two hexamers together in a very stable dihexameric form. And this stable dihexameric form is actually key to understanding why insulin degludec can be combined with rapid-acting insulin aspart, and also why it can be combined with a GLP-1 analog such as liraglutide in a stable co-formulation. So in the cartridge we have, or in the formulation, we have insulin degludec present in this very unique dihexameric form. But what happens, as I told you, when there's no phenyl present, as at the site of injection, is that there will be a rapid release of phenyl, and then you will have the hexamer open in both ends. It changes to the T6 conformation, and that allows now the fatty acid tails to reach into the neighboring hexamer at any stage, at any part of the molecule here, and therefore you have the hexamers now linking up as multi-hexameric strings, the length of like 1,000 to 1,500 hexamers, and the width of just one insulin degludec hexamer. So that happens at the site of injection spontaneously by the release of phenyl from the hexameric substance. Now that wouldn't be really useful for prolonging insulin action if you wouldn't have a release of the insulin degludec monomer for absorption and action. And that takes place gradually over time because zinc is now released from the central cavity there, as you can see here. So over time, a time span of hours, we have a release of zinc from the ends of the hexamer. And by releasing zinc from the end of the hexamer, we have the release of the insulin degludec monomers, can be quite quickly absorbed through the capillaries at the site of injection, and then reach into the interstitial fluid to activate the insulin receptor. And we can show also in the spectroscopic analysis that when zinc concentration is reduced from 6 to 4 to 2 to 0, we have a change of self-association state from the multi-hexamer to the monomer as an illustration of the release of the biologically active species. So we have engineered into the insulin molecule with insulin degludec unique features that allows a new mechanism for prolonging insulin action, and also will allow co-formulation with insulin aspart and with liraglutide. We can visualize formation of the insulin degludec multi-hexamers with electron microscopy. That's done here in an in vitro setting, in a test tube. We can do that also following injection into pig skin. So still these are soluble, fully soluble insulin degludec multi-hexamers. It's just because of the magnification that you get with electron microscopy that you can actually visualize them. But still in solution, in contrast to what we see with NPH insulin or with the precipitate of insulin glargine, where you have formation of large precipitates at the site of injection, as illustrated here with glargine, or with the NPH insulin. We do believe that that feature, that insulin degludec stays in solution, is actually important for reducing variability of absorption of this particular insulin analog. But as I told you, there is, because of this new hexamer substance, there is a possibility to make a co-formulation with rapid-acting insulin aspart into the product that we call IDEC-ASP, or will be a brand name Rhizodec. And let's look at that particular formulation, why that is possible. Well, that is possible because, as I told you, insulin degludec will be present in the formulation in a dihexameric form. And because this dihexamer is so stable, there will be no incorporation of insulin aspart monomers into this type of dihexamer once it has been formed. Rather, insulin aspart, as illustrated here in the orange color, will take up the R6 conformation in the presence of phenyl, and there will be no interaction between the two in the formulation. So we can have a stable co-formulation with no interaction between the components. We can visualize that with size exclusion chromatography, in which you can see that we have distinct peaks for the insulin aspart and the insulin degludec components. But once injected, of course, degludec again undergoes a conformational change, and we have formation of the multi-hexamer. If we can get it to work. Which we cannot. Is there any alternative to this? If I can go back to the IDEC-ASP formulation. So this was IDEC-ASP. And in the formulation, as I showed you, you have the dihexamer of insulin degludec, the hexamer of insulin aspart, in distinct forms, as illustrated here by size exclusion chromatography. At the site of injection, there's in the spontaneous formation of the insulin degludec multihexamers that goes on following release of phenyl. And then following release of phenyl from the insulin aspart hexamer, there's a very rapid dissociation of the aspart hexamer into the monomers, illustrated there in orange. And that insulin aspart monomer, of course, is very rapidly absorbed and undergoes then a receptor interaction and internalization following that. We can also visualize with size exclusion chromatography that we have the distinct formation of insulin aspart monomers out there to the right, and the insulin degludec multihexamer out there to the left following injection. There's also an option to combine with liraglutide because of the stable dihexameric form of insulin degludec into a product that we call IDEC-Lyra, and there will be more presentations in this conference and also in this next couple of sessions on the clinical properties of IDEC-Lyra. Why is that possible? Again, it's possible because insulin degludec forms these dihexameric stable structures, whereas liraglutide, many of you may not be aware of that, but it's actually present in a heptameric form with 7 liraglutide molecules coming together via the fatty acid side chains in the liraglutide molecule in this heptameric form as illustrated here in the magenta color. So in the formulation, we have degludec present as dihexamers and liraglutide present as heptamers. We can again show that by size exclusion chromatography with liraglutide there in the middle and the degludec dihexamer with a little more molecular weight. Following injection, what happens is again that we have a formation of the multihexamer string of insulin degludec, whereas liraglutide stays in a heptameric form also following injection and acts for a 24-hour period, relatively similar to the action profile of insulin degludec, but with a different mechanism of prolongation. Again, that can be visualized by size exclusion chromatography, now with liraglutide staying as heptamers there in the middle, and insulin degludec now moving to the left as an indication of a larger molecular size of the insulin degludec multihexamer. So much for the mechanism of prolongation of degludec and liraglutide and the option to combine. Now let's look briefly at the molecular pharmacology of the insulin degludec molecule. Of course, we want to preserve efficacy and safety, so we also want to retain the receptor binding properties of the insulin molecule. And because there was no amino acid substitution in the insulin degludec amino acid backbone, it's also recognized by the insulin receptor with the same selectivity and with a similar affinity as human insulin, as illustrated here in the top. Following receptor binding, the insulin receptor is activated by degludec, as illustrated here with the 2 classical pathways. First, the AKT or the PKB pathway, and then in the bottom there, the ERK or the MAP kinase pathway, as you can see, activated to a similar extent by insulin degludec as with human insulin. So the molecular pharmacology and the receptor action is similar for degludec and human insulin, and so is the metabolism that the degludec molecule undergoes following receptor binding. So what happens here is that following binding to the receptor degludec complex is internalized into lysosomes, and you get a separation of the degludec molecule from the insulin receptor, and the receptor is then recycled to the cell surface to receive another insulin molecule, whereas the degludec backbone undergoes degradation exactly like human insulin within the lysosome. So there is a proteolytic cleavage of the insulin degludec molecule. And following that proteolytic cleavage of the insulin degludec molecule, there is a full oxidation of the molecule to carbon dioxide and water, whereas the amino acids can be reutilized in the biochemical processes in the cell. And that normally works also in this animation, but today we have some severe problems with these various devices. So we'll see if we can get it back on, just to show the last slide. So we had the signaling, we had the internalization, and now we have the degradation of the insulin degludec molecule, as you can see here, a cleavage at position B24, B25, B26 by proteolytic enzymes, then a full oxidation of the fatty acid component, whereas the peptide backbone can then be degraded into the amino acid components that can be reutilized in the biochemical processes in the cell. So the same metabolic pathways are followed for degludec as for human insulin. So in summary, what do we know about insulin degludec? I hope that you can agree with me that the mechanism of prolongation that we have for insulin degludec is a unique mechanism. It's composed of formation of dihexamers in the pen, multihexamers at the site of injection that allows for a very stable preparation that provides a slow release of insulin degludec as a basal insulin over more than 24 hours. This stable formation of insulin dihexamers allows combination with insulin aspart and also with the GLP-1 analog liraglutide in a stable co-formulation with retention of the individual pharmacokinetic profiles. The action at the receptor, so the molecular pharmacology of insulin degludec, and also the subsequent metabolism of degludec is equivalent to human insulin, so we preserve efficacy and safety of the molecule. And taken together, this design of insulin degludec provides an ultra-long duration of action and a flat pharmacokinetic and pharmacodynamic action profile with unique options for combinations, and you'll hear more about the clinical profiles of those various combinations and of insulin degludec itself in the following presentations. Thank you very much for your attention.
You managed to summarize 25 years of research, but also tolerate the vagaries of the AV system.
Yeah.
So well done.
Thank you.
Well done. So ladies and gentlemen, you can see we're having a little bit of trouble with our technology, but then life wouldn't be so easy if everything went wonderfully well. So just bear with us. We are trying to resolve these issues. I also need to just remind you about how you can contact us, how you can send us your questions. In your packs at the back, you have some question forms, question sheets, question cards that can be ripped out. And if you just write your questions on there, we will have one of our hosts come along and pick that up. So now, so far, we've set you a story. We've introduced you into the story by saying current preparations could do with some improvement and what you and I would desire in terms of a clinical emphasis for a basal insulin. You've heard very elegantly from Peter about the bioengineering behind peptides to deliver and develop some of these insulins that you and I can use. So we now need to move on and to see if these laboratory-based wonderful proteins can fulfill the requirements that we have set. And in order to do that, it's a great pleasure to introduce my colleague Thomas Pieber, Professor of Medicine from Graz in Austria, great experience in insulin pharmacokinetics to describe the clinical profiles of the new long-acting insulins. So, Thomas, you need to tell me, in all the work that you've done so far, do you believe it, that the requirements are being fulfilled?
Well, I mean, it's fascinating molecular engineering we have seen, but it comes down to the clinical performance of a product, and that's what I'm going to review for you.
Great. So just use that one.
Okay, Jiten, thank you very much. Welcome. Let's see if I can handle the technical challenges for my presentation. What I'm going to do now is, you have just heard very nicely how molecular engineering works and what we can do now with these new molecules, and I want to focus a little bit how this new long-acting insulin performs in clinical testing, so I'm reviewing clamp data with you. I'm also looking into the fact that we can combine those two analogs now, the rapid-acting insulins and the long-acting insulins, and also we'll briefly look into the concept of combining a basal insulin analog with a GLP-1 GLP-1 receptor analog. So when it comes to the new long-acting insulins, there is insulin degludec, and I think it's quite convincing that we have now a molecule that's unchanged. The amino acid backbone is completely unchanged. It's human insulin, but it has this prolonged action. And of course, we want to see how that performs in clinical testing. So the first most important feature, I would say, is the long duration of action. So you can measure that in pharmacology as the half-life of a substance. And Tim Heise did this study, and he was comparing the half-life of insulin degludec and was comparing it against insulin glargine. And he did that in very many different concentrations, ranging from 0.4 up to 0.6 and 0.8 units per kilogram body weight. And you nicely can see that the half-life of insulin degludec is twice as long as that of insulin glargine. So glargine has a half-life of around 12.5 hours, and insulin degludec has now a half-life of 25 hours, so it's beyond the 24 hours. And I think that's quite smart, because when you want to have a full coverage of a 24-hour period, you of course need an insulin that's beyond those 24 hours. Like any basal insulin, when you start therapy, you have to get into steady state, and this slide shows you for both type 1 diabetic patients as well as type 2 diabetic patients that we're achieving steady state on the third day after initiating the treatment. Let's also see what this longer half-life means when it comes to insulin fluctuations. The maximum levels and the minimum levels, the peak to trough relationship. And if you look at a basal insulin analog with 12 hours, like insulin glargine, you see fluctuations up to 50%. And when you take an insulin with a longer duration, like insulin degludec, you all of a sudden have a much flatter profile with reduced changes between your peak levels and the lowest levels you have within a day. So at the end, as a patient, you should have much more stable insulin levels, and you should have indeed the reduced variability that was one on the wish list of our chairman today. So a flatter profile when you achieve steady state. And is that true? Well, again, a clamp study performed by Tim Heise. He was testing this 0.4, 0.6, and 0.8 units of insulin degludec per kilogram body weight, and you can nicely see these flat profiles you have in steady state. So there's no peak. It's flat, and if you increase the dose, you just increase your insulin action in a very nice dose-response relationship. It's perfect, and you really can titrate any of your patients now to complete stable insulin levels for your basal insulin therapy. We have performed that study. You even can concentrate the insulin in this new formulation. You can, for example, increase it to 200 units per ml, the same profiles. And this is of advantage, of course, if you have to treat obese people that need larger doses, so you can reduce the volume of your injection by increasing the insulin concentration with insulin degludec. So let's look at another clinical consequence. For example, what happens if you miss a dose? Is there a difference with the traditional basal insulins, like with half-life of 12 hours, like insulin glargine, and this new one? Well, what happens if you miss a dose? If you use insulin glargine, you can see that if you forget to inject one insulin dose, your levels fall down to almost 30% of the maximum insulin levels, and again, you need 3 days to get back to steady state. When you take insulin degludec with this half-life of 25 hours, you see if you just miss one dose, there's a drop of insulin, of course, but it's much less, and you are achieving the same stable insulin levels much easier with this longer duration of action. So when it comes to those mishaps that our patients have once in a while, if they forget to inject insulin or if they don't want to use the insulin on a particular day, with this long duration of action, you have a particular advantage. Another feature will be around flexibility. For example, it's not always feasible for our patients to have an insulin injection exactly every 24 hours. Let's just imagine they're going out for dinner, like you might do tonight, and they go to bed later, so they might want to inject their insulin later, or they want to sleep longer, let's say on a Sunday morning. So what happens with an insulin with a half-life of 12 hours, if you just shift your injection time by 8 hours, and you can see again you have this drop of your insulin levels, and then you almost overshoot when you come back to your 24-hour insulin injection scheme. Whereas when you use insulin degludec with this much longer half-life, you can see there's almost no change in your insulin concentration if you shift your insulin injection time. And this box shows nicely that the difference for insulin with a half-life of 12 hours is between 30% and 120%, whereas for insulin degludec, an insulin with a half-life of 25 hours, it's just between 80 and 100%. So there's another major advantage in daily life for our patients, and this is the increased flexibility. Finally, the last part of my pharmacology insight of insulin degludec is the variability. And again, Tim Heiser did a very nice study. He compared insulin degludec against insulin glargine and was measuring the variability by injecting the insulin on 3 different days and measuring the insulin action. And he did split the full 24 hours into 2-hour intervals. And what you see here is that throughout these 24 hours, the variability you see with insulin degludec is low, and it's the same throughout, as you would expect for this very flat profile. Whereas when you look at insulin glargine, you see a huge change in variability, mostly related to the peak action you have around 8 to 12 hours with insulin glargine. So there is a 4 times lower variability with insulin degludec compared against insulin glargine. So in summary, this new insulin degludec has a half-life beyond 24 hours and a duration of action, as you have seen, up to 42 hours. It really gives you a flat and stable glucose-lowering effect and a significantly lower variability than any other basal insulin at the market, on the market right now. And this new insulin might facilitate now the titration to lower fasting plasma glucose targets to achieve normoglycemia easier. And of course, it has the potential of dosing flexibility, and this would allow to adapt to daily life with insulin degludec much, much easier than you can do with any other insulin preparation. And this, at the end of the day, should lower the risk of hypoglycemia, and it also should lower the risk of hyperglycemia in the clinical trials. So these are the pharmacokinetic properties of insulin degludec, and you have heard by Peter just before that these unique molecules now allow that you can combine them. So there's the idea now to combine long-acting insulin with a rapid-acting insulin like insulin aspart. Why should we like to do that? Well, when you just imagine a patient, a type 2 diabetic patient on basal only, and if you have to intensify therapy, both patients and physicians sometimes stay away from that because they consider it a burden to increase, to intensify the insulin therapy by increasing the number of insulin injections. Another concern is the risk of hypoglycemia, and multiple injections per day are perceived as a burden of the disease and of course might limit the adherence to therapy. So there's the idea that if you can co-formulate those 2 analogs, that could be an advantage in daily insulin therapy. And you have seen that it's possible, so both molecules stay on their own, and of course there's the idea to mix them now in a traditional relationship of 70% of a basal insulin and 30% of a short-acting insulin aspart. And the interesting thing is that when you do that, you can maintain both properties of both insulins. So you have a flat profile with insulin degludec, and you add on that the short-acting profile of your short-acting insulin analog insulin aspart. So when you inject that once daily for your main meal, you have this peaked action for your of your aspart, and you have the prolonged action, the flat profile from insulin degludec without any shoulder and any other problem, as you might see with premixed MPH insulin, for example. And if you go to twice daily, of course, you have the same profiles maintained. So you can really provide your patients with a flat basal insulin and offer a prandial insulin peak, as you might need in some of the patients. So in summary, insulin degludec can be combined with insulin aspart. They are both soluble in a combination product. And insulin degludec and insulin aspart, the individual characteristics are preserved in these formulations, the ultra-long profile of insulin degludec and the rapid acting of insulin aspart. And this might help us for treating our patients, especially if you want to intensify therapy, because you can provide them with one injection with both the meal-specific short-acting and the 24-hour basal insulin with low variability. So using this pre-mixed insulin once or twice daily could be an alternative for basal bolus regimens, but that, of course, has to be clinically tested. So finally, I want to move into this very new and attractive idea of combining a long-acting insulin with a GLP-1 analog. And of course, there are different combinations available. One is insulin degludec in combination with liraglutide. So we have seen by Peter that we can co-formulate it. Insulin degludec and liraglutide both have a 24-hour duration of action. So with a once daily injection of both different molecules, we might be able to achieve better glycemic control by using the complementary pharmacological properties of insulin and GLP-1 receptor agonists. So what could be the benefits? Well, there's the chance that we further can improve the glycemic control. There's the chance that the GLP-1 receptor analogs with their weight benefit in combination with insulin still maintain this weight benefit. And there's the chance to see a reduced risk of hypoglycemia. But again, I agree with you, this needs to be tested in clinical trials, and I think it's quite exciting that today we will see the first results from those clinical trials. So dear colleagues, ladies and gentlemen, in summary, the new long-acting insulin analogs and the combination products have recently become available or are under development. I think that's very exciting times right now. We are in the middle of it. The clinical profiles of these therapies may represent an advance over the pre-existing treatments with more physiologic pharmacokinetics and pharmacodynamic properties. And these new therapies could contribute to improved glycemic control, less hypoglycemia, and increased convenience for our patients. Thank you very much for your attention.
Thank you very much, Thomas. Well done, well done. You seem to have resolved our technological issues as well. So ladies and gentlemen, we think we've resurrected the AV system and the technology glitches have been removed. So thank you very much for your patience with us. So what I'd like to just do is to summarize where we are now for a moment. We started off by an introduction and we said we'd like a basal insulin which was longer, flatter, less variable, which allowed flexibility for the patients. So you've heard some really clever peptide engineering to demonstrate the development of such a molecule, and you've now seen the clinical profiles of the molecule and the ability to combine these new molecules in one administration device. So we now need to move on and ask the question that we posed, which is, will the new devices reduce the rate of hypoglycemia, which is, of course, the key issue for the patient? And to try and answer this question, we have Athina Philistimakis from San Diego, big researcher, loads of trials in terms of pharmacokinetics and clinical studies in type 1 and type 2 diabetes. So, Athina, have you been impressed by the data that you've seen around the hypoglycemia reduction with the new preparations? And are you going to convince us?
I have been impressed, and you know, in my role where I oversee 5 hospitals and 25 outpatient clinics for diabetes services, the #1 question for me always is safety. So I have to be convinced that the safety is there before we make recommendations.
Well, convince us.
Great, thanks, Jiten. Good morning, everybody. It's a pleasure to be here today. Today I'm going to go over some of the first studies in the phase 3 that have been done with degludec, both in type 2 and type 1 patients. We'll also look at the meta-analysis that was done and a post hoc analysis, and then I'll finish up with a few special situations that people might have questions about and wonder, is it really okay and safe to use the new insulin degludec? So let's get started. Maybe just as a background, show you, as all of you already know, what really contributes to glycemic control. And we know there's fasting plasma glucose as well as preprandial glucose throughout the day. There's postprandial glucose that contributes, as well as hypoglycemia, which can occur anytime during the day, but many times can occur nocturnally and can be very dangerous. So, is hypoglycemia a problem? Are you worried about it with diabetes therapy? And this study looked at that question and found that out of the top 4 drugs that are dangerous, cause dangerous situations, 2 of those are glucose-lowering therapies. And when they looked at the overall results of hospital emergency room hospitalizations, 95% of all endocrine emergency hospitalizations in people over 65 years old were caused by hypoglycemia. And you might think about some of your patients, your 70 or 80-year-olds that maybe entered that emergency room with a fall, broken hip, that had to stay for a week or 2 to get taken care of. So I think it definitely is an issue, and it's something that we want to avoid, if possible, while optimizing glucose control. Just to remind you, across all the studies you'll hear about today, the same definition for hypoglycemia was used, and this is how it was defined in those studies. If someone had symptoms of hypoglycemia, or during measuring at very standard times, if there was an unresponsive event, just like in all definitions of hypoglycemia, that was considered severe hypoglycemia. If they were able to measure, they made the measurement, and if that measurement was less than 3.1 millimoles per liter, that was considered a confirmed hypoglycemic event. If that event occurred between midnight and 6 a.m., that was considered a nocturnal hypoglycemic event. So these are the 2 studies that I'll discuss. You saw these earlier in that framework that Jiten showed you of all the studies that have been done to date. I'll also show you the results from the extension studies which were done in both type 2 and type 1. So this is the type 2 study. This is your group of patients with type 2 that are on oral therapies and not optimally controlled. So you're going to think about adding a basal insulin to this group. Insulin naive, and they were randomized to receive either insulin degludec once a day or insulin glargine once a day. They were maintained on metformin, and then if they were on a DPP-4 inhibitor, they were allowed to maintain that as well. Those were the only medications that they were placed on. They looked at the results at 52 weeks and then did an extension for an additional 52 weeks. There was a 1-week washout time period, you'll see that in between, in which insulin autoantibodies were measured to see if those were elevated or not. So what were the results? First, in the primary outcomes, because these were treat-to-target non-inferiority trials, there was a very distinct approach to maintaining your glucose levels at target. And so, as expected, your hemoglobin A1C levels dropped nicely and were also equal between the 2 groups. Fasting plasma glucose, also when you looked at it, dropped nicely, although interestingly, the insulin degludec group had a slightly lower fasting plasma glucose. And as you'll see throughout the day today in studies that are shown to you, that's a characteristic that has appeared in all the studies. So because you're able to maintain equal glycemic control between groups, this is nice because it allows you to then look at some other characteristics that you might want to differentiate the 2 insulins with. So in hypoglycemia, what happened when you maintained glycemic control equally between the 2 arms? And what you see here is that for overall confirmed hypoglycemia, that with insulin degludec you had a lower rate of hypoglycemia, numerically, although not statistically significant in overall. And then in nocturnal, which we might consider the more dangerous type of hypoglycemia, because you're not always aware of this hypoglycemia when it occurs, you can see a really nice division of the rates of hypoglycemia, with lower hypoglycemia rates with the insulin degludec compared to insulin glargine. And if we look at this from a clinical perspective, how many patients would you need to treat in order to prevent one hypoglycemic episode? You'd need to treat 7 patients for a year. If you treated 100 patients for a year with insulin degludec compared to insulin glargine, you would have 15 fewer episodes of nocturnal confirmed hypoglycemia. Okay, let's turn briefly now to the type 1 study. Basal bolus, all our patients with type 1 are usually on basal bolus therapy. And they could come from any type of basal bolus therapy and get switched to either insulin degludec plus insulin aspart or insulin glargine plus insulin aspart. Followed again over the 52 weeks, 1-week washout, and then an extension period for an additional 52 weeks. So looking at these results, hemoglobin A1C, again between the two groups, nice drops, equal between the two groups. Fasting plasma glucose, again drops, slight elevation during that washout period, but then as soon as they were maintained back on their doses, came back down again. And again, you see that slightly lower fasting plasma glucose in these groups. So before I turn to the full hypoglycemia across both the initial time period and the extension, I think it's worth just showing you some of the initial hypoglycemia outcomes to look at maybe some of the mechanisms that might have contributed to these results. So in the first 52 weeks, the rates of hypoglycemia between both groups ran pretty parallel, you can see in the lines here. What happened, though, in the first 4-week time period of this study? And if you think about patients came from any type of basal bolus therapy I told you, and it could have been BID basal therapy, switching to the once-a-day basal that they were placed on. And following label, if they were on BID, which approximately 30% of the patients were, their dose was reduced by 20% within that first conversion time period. You can see that they very quickly went back up. They were titrated up in order to achieve the control that was necessary, but they did start out with a lower dose of glargine when they were entered onto that arm. So what you see in this lower graph is what the rates of hypoglycemia were, confirmed hypoglycemia. And if you look in that first 4-week time period, they actually had lower rates with the insulin glargine in that first 4-week time period. If you look across the remainder of the time, the rates of hypoglycemia were identical, but certainly having lowered that dose may have contributed to the lower rates in the first 4 weeks, and that's part of the intricacies of doing a clinical trial. You don't know exactly what's going to happen, and this is what was found at that time. So you might redesign the study the next time you do this to have similar approaches for both your arms. So keeping that in mind, what did happen with confirmed hypoglycemia across the entire extension period? You can see that those parallel lines continued and almost look like they actually do cross over in the extension period and begin to reverse. No difference in hypoglycemia here. What about if you look at nocturnal confirmed, again, that dangerous time period? And what you see here is actually a reduction. So a significant reduction of 31% in the insulin degludec arm compared to the insulin glargine arm. So again, looking at the interpretation of this, patients with type 1 have many more events than type 2. So here, to avoid one hypoglycemic episode, you'd need to treat only one patient for 8 months to avoid that. And if you treat 100 patients for a year with insulin degludec compared to glargine, you get 158 fewer episodes. So the summary of the extension data, shows that insulin degludec really very nicely improved glycemic control and maintained it after 104 weeks in both type 1 and type 2 patients. And there were statistically lower rates of nocturnal confirmed hypoglycemia in both these groups as well, 42% in type 2, 31% in type 1. Okay, switching to the meta-analysis. You remember I told you that by using the same measurement outcomes for hypoglycemia across all the studies, this then allows you to combine them all and do a very nice meta-analysis at the end of this. So that's what I'm going to show you, is just the initial results, the pooled results, for both confirmed hypoglycemia as well as the nocturnal. So what you see here on the top part of the graph here was the overall hypoglycemia rates. If you take the pooled insulin naive and the pooled type 2, you can see across the entire trial, nice reduction. Pooled type 1, there was 10% increase, but that was not statistically significant and may be explained by that first 4-week time period that I spoke to you about. In the nocturnal, you can see again here a very nice reduction in the pooled insulin naive as well as the pooled type 2, and a reduction in the type 1, which was not statistically significant across the entire time period. But what was done afterwards was something else that was very interesting. Some of the hypoglycemic events that you would expect many times occur in those first 16 weeks as you're adjusting, as you're titrating your insulin doses. And therefore, a maintenance period meta-analysis was also done in which from the 16-week time period onward, they looked at what the rates of hypoglycemia were. And here you can see not only are the rates maintained as before, but they're actually even better. They're improved even more. So in the overall time period for both type 2, there's nice reduced rates of hypoglycemia. In the type 1s, there's that improvement over time that we saw in the one trial that I showed you. And then looking at nocturnal, now all three, both pooled type 1 and pooled type 2, have very nice reductions in hypoglycemia rates. So looking at one more interesting analysis and composite data, because again you had these similar outcomes, you could now combine studies. This is a combined type 2 and basal oral therapy, looking at the combination of that fasting plasma glucose. How low could you get that at the same time not have hypoglycemia compared to degludec versus glargine? And this is, you can see across all the trials that were done with type 2, and then combined in those first two set of bars what the differences were. And you can see that patients with type 2 diabetes on basal oral therapy had an 82% higher likelihood of reaching that fasting plasma glucose level without having any nocturnal hypoglycemia in the insulin degludec arm compared to insulin glargine. So finally, the last few slides are just turning to some special situations you might think about with patients in these situations. So patients with type 2 diabetes that have had it greater than 10 years. We've seen the studies that show that they become a little more sensitive, that they have hypoglycemia rates that begin to equal that of people with type 1 diabetes. So are they more likely to have hypoglycemia when they're using insulin degludec compared to insulin glargine? And this meta-analysis, this post-hoc analysis actually, looked at this and showed that there was no increased hypoglycemia rates, whether they had the diabetes greater than 10 years as compared to the entire population, despite both groups achieving good glucose control. The next situation we might think about is how about exercise? If you're on a longer acting, longer duration insulin, would there be more hypoglycemic episodes with exercise? And this was a patient response, patient-reported outcomes. Patients exercising, did they have any increased events? Did their events occur more frequently than those on insulin degludec versus glargine? You can see here that there was no difference in those responses. This is a patient-reported outcome study, but now a much more formal study has been done. It's going to be presented for the first time here at this conference, and you can see what the results of that are on Tuesday. Finally, the last question you might ask is, if someone has a hypoglycemic episode, if they're on a longer duration of action insulin, would it take them longer to recover from that episode? So this was a very nice study that was done by a few of our presenters that are here today. And what it showed with an induced hypoglycemia during a glucose clamp situation, that if you look at the glucose infusion rate required after that induced hypoglycemia, there was actually less glucose infusion required in the group taking degludec compared to glargine. And the time to recover was less as well. So I think that alleviates some of the fear around that situation as well. So just to conclude, from all the information I showed you, in treat-to-target studies, insulin degludec demonstrated a lower risk of hypoglycemia compared with insulin glargine, while achieving similar rates of glucose control. Both in pre-specified and post-hoc analyses, there were lower rates of confirmed and nocturnal confirmed hypoglycemia with degludec compared to glargine. And there was no difference in the exercise-related hypoglycemia, on the impact, duration, or recovery from hypoglycemia with insulin degludec compared to glargine. So I believe there's an extensive amount of data out there today demonstrating the safety. I would feel comfortable recommending this in my patients, in my health system, and I look forward to seeing how this will be used really in many situations throughout the world. It's been a pleasure speaking to all of you today. Thank you.
Excellent. Thank you very much. Thank you. So, ladies and gentlemen, the story continues and develops. Introduction, demands, requirements of an ideal insulin, bioengineering, clinical pharmacokinetics, and now demonstrating lower hypoglycemia rates. So the other great expectation we have of an ideal basal insulin is the flexibility of administration. And to demonstrate and present the data on flexibility of insulin degludec, we have a great friend of mine, Luigi Mangini from Miami, professor of medicine in Miami, who's going to present data on the flexibility advantages of insulin degludec. Now, Luigi, you heard from Thomas, we have a series of patients, in fact, many patients, who have this mishap, this difficulty with missed doses, delayed doses. Your opinion on this?
Yeah, so I think that maybe instead of calling it mishap, I would say that maybe life gets in the way of diabetes. And this, I think, provides us with a wonderful opportunity of actually looking at whether we can introduce flexibility based on some of the pharmacokinetics and clinical data, and actually have diabetes become part of life.
Great. Well, floor's yours.
Thank you very much. So good morning, everyone. What I'd like to start discussing is exactly that. Do we want to force our patients into our prescription, which often is rigid and fixed? Or do we want to take the opportunity with some of the therapies that we have of actually adapting those to patient needs? So introducing flexibility and adaptability. And what we've seen today gives us an opportunity of possibly doing that. And we know that as physicians, we write prescriptions for patients, and we expect these patients to follow these prescriptions to the letter. But I don't think any one of us is naive enough to think that many of those patients miss doses of insulin, of other medications, mistime these doses of insulin, and this study is actually looking at basal insulin replacement, and actually might reduce doses of insulin, sometimes appropriately, but many times not appropriately. And we also know that adherence has an impact on outcomes. And these outcomes, related to poor adherence, can translate into worsening of complications. So patients do report increased kidney problems when they are also found to be poorly adherent to medications. And there's an increased incidence of mortality, diabetes-related or overall, when these patients are identified as not being adherent to medication. And we also know that with regards to patients with diabetes, over a third of them actually don't take their medications consistently. Not a big surprise, but just as many report not taking their insulin consistently. And from simulation models, we know that missing insulin doses actually translates into higher A1C values. In this simulation model, missing 2 basal insulin injections per week actually increased the A1C values by about 2.2 to 0.3%. So we're kind of in a quagmire because traditionally we have issued the prescription, we have told the patient to follow the prescription, and we basically have not used a very patient-friendly or user-friendly approach. But the professional organizations are actually recognizing that we need to actually adapt diabetes to life, or our prescription to the patient, individualize not only the goals of therapy, but the treatment that we give to achieve those goals. And so with that, we could look at our patient that's scheduled to take a basal insulin injection at 7 p.m., but might have work functions or might have social situations that get into the way of that insulin injection. So that in effect, they might skip doses, they might anticipate doses, they might delay doses. And do we have an opportunity with a longer acting basal insulin of introducing some forgiveness, and in that manner introducing some flexibility so that we can allow the patient that flexibility of either injecting earlier or later and not compromise either efficacy or safety. So this was the concept that led to the FLEX studies. And I'm going to show you 2 studies. I'm going to show you the first study in type 2 diabetes and the second study in type 1 diabetes. And the idea is, well, we have a long-acting basal insulin that we know has a flat, stable, and consistent profile. Could we introduce flexibility in the administration and the timing of that insulin administration and still retain the same efficacy that we've seen with other studies and the same safety that we've seen related to insulin degludec? So patients with type 2 diabetes, not well controlled on oral medications, were randomized to 3 arms. The first arm was insulin degludec given in a flexible format. And I'll show you what that means in a second. The other 2 arms were given the basal insulin at the same time every day. And I'll show you the example of that. So the fixed once daily dosing for insulin degludec occurred in the evening time, the same time every evening. The fixed dosing for insulin glargine actually occurred at any time that the physician prescribed the glargine, but it had to occur at that same time every day. On the other hand, the flexible regimen was an artificial way of trying to introduce a lot of variability between insulin injections. So patients were actually instructed on the first day to inject in the morning, on the second day to inject in the evening, the third day in the morning, the fourth day in the evening, in fact creating administration times of anywhere from 8 hours to 40 hours between basal insulin injection, insulin degludec injection. So not something that I would recommend you try with your patients, but certainly a way of testing the flexibility issue with insulin degludec. So what happened? In terms of A1C over time, you can see that there was no difference in terms of A1C reduction, whether you allowed, actually you forced patient to inject insulin degludec with timing intervals between 8 and 40 hours. Pretty remarkable. No difference compared to once daily at the same time insulin degludec or insulin glargine. Well, what about hypoglycemia? We haven't compromised safety. We haven't compromised efficacy, but what about safety? Athena showed you that there are some advantages to these ultra-long-acting basal insulin in terms of reducing hypoglycemia, especially nocturnal hypoglycemia. Have we completely obliterated that by introducing these wild dosing intervals for insulin degludec? In fact, when you look at the rates of nocturnal confirmed hypoglycemia, there's no statistical difference among the 3 arms. So actually, force flexing insulin degludec did not compromise safety in terms of hypoglycemia compared to taking either insulin degludec or insulin glargine at the same time every day. And actually, there was a lower fasting plasma glucose that was observed in the force flex arm compared to insulin glargine, something that was alluded to by previous speakers. So that's type 2 diabetes. So we've forced them to take insulin degludec in the morning, in the evening, at very wide intervals, and have been able to maintain glycemic control without compromising safety. But as you know, with type 2 diabetes, they have endogenous insulin production. They have some buffer. They have some protection against fluctuations in insulin levels. Well, what about type 1 diabetes, where there's complete insulin deficiency? Can we take this Wild West approach, if you want, do it with type 1 diabetes, and still preserve efficacy and safety? So this was done in the FLEX-1 study, which took individuals with type 1 diabetes on basal bolus insulin therapy, and for the first 26 weeks, randomized them to either this forced flexible approach, similar to what I've shown you with the type 2 study, compared to a fixed degludec approach and a fixed glargine approach. So the degludec and the glargine in the other two arms dosed at the same time every day. At the end of these 26 weeks, what they did is they actually asked patients who were on degludec to free flex, meaning you can take the insulin degludec at any time of the day, just within the 8 to 40-hour interval that had been tested in type 2 diabetes. So kind of reproducing what patients actually would do if you give them an insulin prescription for basal insulin. The arm with insulin glargine, on the other hand, continued injecting the glargine at the same time every day. And what were the results? Well, in terms of efficacy, there was no inferiority for insulin degludec in the force-flexed arm compared to the insulin glargine. Pretty remarkable if you think about a population of type 1 diabetes taking their basal insulin once in the morning, the next day in the evening, the next day in the morning, and so on and so forth. And when you freed up the dosing or the timing of insulin degludec in the free flex format, no difference in terms of efficacy when compared to the insulin glargine arm. Fasting plasma glucose, again, no difference in terms of the forced flex versus the glargine arm in terms of fasting plasma glucose for those 26 weeks. But then when you freed up the flexible dosing for insulin degludec, at the end of the trial there was a slightly lower fasting plasma glucose associated with a free flex approach of insulin degludec compared to insulin glargine. Well, what about hypoglycemia? Confirmed hypoglycemia, confirmed overall hypoglycemia, was no different whether you used a forced flexible approach compared to a once-a-day fixed approach for either insulin degludec or insulin glargine. And actually, free flexing for the extension study, again, did not compromise the risk of hypoglycemia compared to insulin glargine. Now, overall hypoglycemia doesn't seem to be different in many studies, but nocturnal hypoglycemia, especially when these basal insulins are dosed at night, might be an issue. And so that was looked at as well. And in the forced flexible 26-week trial, the rate of nocturnal hypoglycemia was actually lower compared to either of the fixed arms. And when allowed to free flex the insulin degludec for the remainder of the extension trial, that lower rate of hypoglycemia for insulin degludec persisted compared to the once-daily insulin glargine. And if you put this in clinical context, well, what does that mean for our patients? Looking specifically at nocturnal hypoglycemia, what we see is that in order to avoid one episode of nocturnal hypoglycemia, you would have to treat a patient for 6 months in order to achieve that. And if you had a patient panel of 100 patients that you were treating for a year, you could potentially reduce the risk of hypoglycemia by 208 episodes using a degludec approach versus a glargine approach. Pretty significant findings. But I think the most important lesson to learn from these FLEX studies is that we've been able to introduce some flexibility where traditionally there hasn't been any in terms of insulin dose administration. And we've been able to do that while preserving the efficacy of insulin degludec, and more importantly, by preserving the safety of insulin degludec. And I think this is a new paradigm approach in terms of the treatment of patients with diabetes that kind of responds to the professional associations that tell us that we need to individualize our approach to these patients. Thank you very much.
Thank you very much, Luigi. Now, ladies and gentlemen, with that, we come to the end of the first part of our story. We are running a few minutes late, so I would suggest that at this point we break for coffee, which will be served outside, and perhaps you would be kind enough to come back fairly quickly at about 10 past 11. So thank you very much, and see you back very shortly.